Well, thank you, Professor Curry and Dr. Ng, for the very kind introduction. And those two were my teachers in psychiatry also, and I think in front of me here are quite a few of my teachers in psychiatry as well. I'd like to thank also Prof. Kwa for inviting me to speak, and also for providing this very provocative title, Has Academic Psychiatry a Future? It's well, it comes on the back of these survey results, which suggest that academics and administration rank pretty low in what is perceived to be a good psychiatrist. Fortunately, in the survey, they didn't ask, "What do you think about physician neuroscientists?" which is actually the focus of my my area. If they had asked that, perhaps that would even pull apart academics from administration and bring it much lower. So it was a very provocative title, I think, and I feel that we need to talk a little bit about does academic psychiatry indeed really have a future or not? Well, if you were to look at the distribution of the global burden of disease by the WHO projected into the year 2020, neuropsychiatric disorders are going to take up a large slice of the pie, some 30%. And the stuff that we've talked about yesterday, today, about how to improve primary health care, how to improve psychiatric teaching, how to improve adherence to standards of care in psychiatry, all that is extremely important. But is it going to cure neuropsychiatric disorders? The answer is probably no. Is it going to dramatically change the cognitive deficits associated with schizophrenia? The answer is probably no. Is it going to change the progression of Alzheimer's disease? Probably no, right? And so the burden of neuropsychiatric illness is likely to remain very high in the foreseeable future. And I think the only way forward, I guess, would be to better understand these illnesses in terms of brain mechanisms, what is really happening, and to design rational therapies to treat these conditions and to find new treatments that are better than the existing treatments. And we all know that even drugs, don't work as well as they should. And psychological therapies don't work better than drugs either. So has academic psychiatry a future? I think the answer should be yes, because without academic psychiatrists, but academic physicians working together with neuroscientists, with government agencies, funding bodies, with industry, it's going to be very difficult for us to find new treatments to reduce the burden of neuropsychiatric diseases. But there is always a caveat. What is the likely future of academic psychiatry? Now, here are some names of just random pickings of contemporary physician neuroscientists, the captains of industry and academia also. For example, Husseini Manji was the director of the Mood and Anxiety Disorders Program at the NIMH. He's now a vice president of CNS in Johnson & Johnson, one of the largest pharmaceutical companies. Mike Egan, my colleague, is director of neuroscience in Merck. This is I just pulled this out of the Genentech website. Dr. Sheng is now the vice president of neuroscience. Now, this is his CV. And something that strikes me, and I guess to a lot of people, is the long training that is required to be a physician neuroscientist, right? It's 8 years medical training, 5 years basic science training, another 4 years advanced scientific training, 12, 15 years after training, and then, an impressive body of work in the top scientific literature, eventually, reaching the pinnacle of his career. So it's a long road for a physician neuroscientist. So it's not for the faint of heart. There is very big attrition rates. At early in career, everybody is interested in research at a high school level, secondary school level. Everybody wants to be an astronaut or something. But the number of people that actually get down to doing it is very low. Even amongst people who commit 3 to 4 years of training in developed countries, some 50% do not continue on in academia, okay, and go on to mainly clinical care, although they still do have a small parts to play in academic work. So it's big attrition, and so why bother, right? Now I'm just gonna give some personal perspectives now, and it can be a very interesting journey. At the end of the day, it's the journey. It's not so much the results. It's a long journey. You've got to enjoy it. It can be exhilarating. It can be depressing. It's ups and downs, depending on what you find. But without a doubt, in the academic world, we get to ask a lot of interesting questions. We learn a lot. We learn a lot from patients, but we also learn a lot about a lot of science and a lot of math. We need to learn a lot about clinical medicine, not only definitely what's in the textbooks, because there's no way you can go beyond what's in the textbook if you don't know already what's in the textbook. We learn a lot about management, managing budgets, managing non-clinical staff, scientific staff, and learn a lot about yourself, people in the world, mainly because academia is so interconnected and so global nowadays. And so we have a privilege of having a sort of a high-octane global network of mentors, collaborators, friends, and all working towards a rather lofty goal, which is to find new knowledge about new disease mechanisms and treatment. Contributing the need to contribute to the scientific knowledge base and not just parasitize from it. Another view that I hold is that, well, it's actually a win-win situation. When you contribute to the scientific knowledge base, you also stimulate other people to do the same. And then a positive feedback loop is generated. And that's where all the sparks will fly, creativity will fly, great new ideas will come up, and hopefully some of these will lead to new treatments. And certainly, academic medicine is one very good way to attract talented students and talented scientists into psychiatry because of its strong, solid base in the neurosciences and in the mathematical sciences. So how does one get involved in some of this stuff? I started my clinical work here 10 years ago. There's no, research is done for the most part in the evenings when you're on call at 3 a.m. You don't want to sleep, that's the way it's done. There was no additional funding, but something that we wanted to do was to sort of ask some interesting questions and try to take advantage of what is available. And what was available at that time was that, well, in Singapore, all the students at age 12, they take the same exam, so you can study that. And all the students at age 16, O-levels, they also take the same exam. So we were interested to study cognitive deficits associated with schizophrenia. And we wanted to study cognitive deficits before onset of illness, which was more interesting because everybody knew that after illness onset, there were already cognitive deficits. So we studied patients with schizophrenia at age 18 to 20 and compared that to a very highly matched group of healthy individuals. And we found that 5 to 6 years before the onset of illness, there was a significant deterioration in cognitive deficits, in cognitive performance, particularly in mathematics-related subjects, 5 to 6 years before the onset of illness. And that was very interesting, which would suggest that the treatments for these cognitive deficits, well, you would have to start thinking about that early on in the illness, perhaps even before illness onset. And in research, we always dig deeper and deeper. So what's the next step to better understand some of these cognitive processes? And so we move on to look at working memory, which is a executive process that is central to mathematical operations, to most cognitive operations. And we wanted to study it in the context of MRI. MRIs are widely available in many countries. It isn't difficult at all to modify an MRI scanner to be able to do functional neuroimaging. So we managed to collaborate with some people who are very experienced in doing that. And again, this work is not funded. It is not done. It is also done, during one's free time. But we studied this fMRI paradigm in patients with first episode schizophrenia and compared it to healthy individuals. Now, I'm not going to go too much into the science here. I'm just going to touch on the spirit and the thinking and the motivations behind some of this work. But basically comparing the brain activation networks of patients with schizophrenia versus healthy individuals, we found that there are big differences between the way patients engage brain networks compared to healthy individuals. Patients with schizophrenia tended to engage a greater amount of prefrontal cortical activation, which is the front part of the brain, and they use a larger network of brain areas. They're more inefficient compared to healthy individuals who are performing the same task to the same level. So digging even deeper into what these brain activation signals mean. Now, these brain activation signals come out from blood oxygenation changes that the MRI picks up. Blood oxygenation changes do not necessarily reflect actual brain activity, neurotransmitter levels, brain synapses between neurons, and so on. They are indirect correlates of that. So what is happening at the level of brain synapses, brain neurotransmitters, brain receptors, and so on? So how do we push MRI to tell us more about the story behind some of these activation changes? And so this was work mainly done in the US, and we then have to link with basic biology. And a lot of basic biology tells us that dopamine is a key modulator in working memory. Well, dopamine is also a key modulator involved in schizophrenia. So now there are numerous dopamine receptors. Dopamine D1 receptors have been shown a lot of basic experiments and neurocomputational models to be engaged in signal-to-noise differentiation in cortical networks or in brain networks. They increase relevant signal and suppress irrelevant noise. Dopamine D2 receptor signals from the striatum and engages in updating information in cortical networks. So it opens the gate to allow new information to flow into cortical networks. So D1 and D2 functions. Can we study D1 dopamine functions in the brain? How do we do that? We can do that by increasing or decreasing amount of dopamine in the brain. How do you increase or decrease dopamine in the brain? You can give a person drugs. That's one thing. You can use radioactive tracers. But we decided to look at a gene that breaks down dopamine in the brain. Catechol-O-methyltransferase is a gene that breaks down dopamine in the cortex. And people have two different versions of this gene. Some people with the VAL allele of COMT have less dopamine in the brain because that version of the enzyme has got more activity. It's more active. Therefore, it breaks down more dopamine, and therefore they have less dopamine in the brain. Whereas people with MET alleles have increased amount of dopamine in the brain. So we can then study two different groups of people, those with this allele versus those with the other allele, and then look at how their brain activation patterns change. And we can then study these different processes and show that basically COMT dopaminergic function impact upon working memory brain function in a way that can be very lawful and predicted by basic science, therefore linking basic science to the human system. These basic science studies have all been done in animals and in cell cultures, but now we show that it reliably predicts what can happen in the human system, and therefore it's a model for translational research. Now, I'm not going to go too much into a lot more that we've done. We've worked on neural simulation models. We can bring in a lot of this data into differential equations and run them through and look at how they process noise in the system. We have looked at how some of these genes interact with other NMDA systems. Dopamine doesn't act alone. Dopamine acts together with glutaminergic systems. And when we look at glutamate-related genes and how they interact with COMT, and we show very lawful predictions where the COMT allele that is associated with reduced dopamine together with the GRM3 risk allele associated with schizophrenia predictably gives rise to the most inefficient prefrontal cortical brain function that during a working memory task. We have gone on to look at intracellular signaling mechanisms downstream of the dopamine D2 receptor, and also show that in genetic variants which have resulted in reduced expression of AKT1, which is also what happens in schizophrenia, these subjects have got inefficient prefrontal cortical brain activation patterns, and this is in interaction with COMT genotype. Now, we can go on and on about the science that appears when one digs deeper and deeper into these systems. But what does all this mean? I mean, is it worth it or not? One could think about it in several ways. Now, this work with GRM3, where we found this lawful prediction, we published it in 2007, and just 3 months later, this study came out. This is a study on a GRM3 targeting drug that Lilly is developing. It is effective for symptoms of schizophrenia in a fairly large phase 2 clinical trial. Now, this is the most advanced new molecule in clinical trials today as a new treatment for schizophrenia. There are a lot of other molecules, but this is the one that's most advanced. Based on our results, we would predict this type of result, and also we would predict that GRM3 related drugs together with dopaminergic drugs could potentially interact together to influence and improve working memory brain function. Okay. The work on AKT1 follows a similar story. Thought leaders in the field believe that this molecule could potentially be useful in the effort to explore more avenues to find new potential treatments for schizophrenia and related disorders. AKT1 has been well studied in cancer and cardiology. There are a lot of drug targets out there that hit on AKT1, and therefore a lot of drug companies are now looking at this molecule in relation to potential new clinical treatments for schizophrenia. Well, I mean, we could think about these findings as impacting new treatments. Another way of thinking about it is, well, at a more clinical level, I mean, we are very grateful to a lot of patients that go through these clinical trials. We have to look after these patients. Some of these patients have to stay in the hospital for 3 to 6 months. Sometimes we have to take them off medications. We have to look after them sometimes with when their symptoms increase. And we're very grateful to patients who come forward to do these clinical trials. But what is very gratifying also is that these patients are also very, very grateful to have taken part in these clinical trials. That's the experience in Singapore when we did some of the work, and also in the US. By taking part in these clinical trials, we also sometimes provide patients with the opportunity to view their illnesses in a very different way. They are not just receivers of care, but these patients have now been empowered in a way that they now contribute to new knowledge that may not benefit them, but may benefit generations of other patients with this illness, and therefore using their illness to contribute to new knowledge. I mean, that's a very powerful way of reframing the way they think about their illness, and it's really gratifying to see that a lot of patients do appreciate that, as well as their family members. So, it is very gratifying doing some of this academic work. Now, I'll just sort of close off with this passage from Liz Nabel. She is the current president of the American Society for Clinical Investigation and the director of the National Heart, Lung, Blood Institute. She's a noted cardiologist and clinician scientist. When my husband Gary and I left the safe haven of our postdoctoral fellowships in Whitehead Institute and the MIT and Brigham's Hospital to join the faculty at University of Michigan, we entered uncertain territory. Well, this is the first advice that she gives to young people. Go do training in the top places in the world if you can. And then they were lured by strong recruiting. Then go to places that welcome you, that provides you with an inspiring academic environment, supportive mentors. That is what makes all the difference. Okay, that's what she writes. The chair of the department is very important. He was energetically building outstanding academic programs. He surrounded the department with a lot of very talented colleagues. And if you look at the names here, a lot of them are big names in the field. Francis Collins, for example, was one of the key people that was involved in the successful completion of the draft human genome sequence. Okay? And these people in the young faculty were all in their mid to late 30s. Again, remembering how long it takes to train physician neuroscientists or physician scientists in general. They were free to pursue their research ideas. Sparks erupted. They were energetic, creative. They fed off each other's vitality. And the head of department understood the value of fostering an engaging and intellectually vibrant academic culture, standing back to let us pursue our creative interests, but stepping in when we needed support and resources. So with that, I'd like to thank a lot of my teachers here and a lot of my mentors and collaborators and friends in the US, funding agencies, students, past and present. Thank you very much.